In this lab, our group was the spectroscopy group. This means we were responsible for preparing and analyzing the fractions that were separated by the FPLC team. We were responsible for identifying which of the samples contained MGH protein. Through the use of the microplate reader, we were able to analyze our samples we created. We were then able to pull the samples that contained the most protein. Our standard curve was created using the data collected. However, in order to obtain the straight line on the next slide, you will see a new standard curve without the 1500 and 750 concentrations. The problems with the 1500 and 750 concentrations could be due to a reading error or a contamination error. As you can see, without these points added to our standard curve, the R-squared value is closer to 1.
From the absorbance at 595 data, you can see that there are peaks indicating the presence of a protein that is stained with Kumasi blue. And the absorbance at 492 is the ideal absorbance to analyze the desired protein, that's in orange. The first spike in the GFP data shows absorbance points of proteins that are similar to our protein of interest. They have histidine-containing proteins and will bind to the column in the same way that our target protein will bind. And this method cannot separate other histidine contaminants from our protein. The second spike in GFP data shows the presence of our desired protein.
Fractions 5 through 9 have a spike due to contaminants. The absorbance then decreases, then spikes at fractions 21 through 24, signifying the release of our MGH protein from the column. It is very unlikely that our MGH protein would be fractioned off so early in fractions 5 through 9, so it is most likely due to a histidine-containing contaminant. Using protease may interfere with yield, as it could separate our Mg protein from histidine, allowing broken histidine molecules to stick to the column instead of our Mg protein, causing a major discrepancy in data.
This table goes into depth discussing the total protein concentration and amounts based on data from the Bradford assay determined by the standard curve. To solve for the amount of MGH present, we will use this equation right here from the standard curve, where Y will be our absorbance, and we will be solving for X. So we're going to plug in the absorbance readings from each fraction right here and do a little bit of algebra to solve for X. And these are the X values from all of those calculations. And to find the absolute concentration for each fraction, or the amount of protein per fraction in micrograms, we're going to multiply all of these numbers by 3, because we need to account for 3 milliliters of fraction for each of these. So you would just multiply by 3, and that's how you get to these numbers, which tell you the amount of protein per fraction. So to determine the absolute amount of MGH protein present in our mixture, you're going to take the sum of all of these proteins, and then that is what you should get, which would be 73.125 milligrams. The total amount of protein present in the pooled fractions will be used to find the percent yield, which is done by comparing to our theoretical yield and multiplying by 100. This allows us to see which fractions certainly contain our MGH protein, which are shown here as well. You'll see 20 through 24 right here. And we also believe that there may be some present here, but that could also be high readings due to impurities.